Biopharma presentations. I am Xavier Gutierrez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. Now let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. Please address each question to a specific speaker. We will answer as many questions as we have time for at the end of the presentation. And if you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I now present today's speakers. First, we have Dr. Mimi Roy, Executive Director of Analytical Science at Biopharmaceuticals, presenting on the influence of mass spectrometry in development of host cell protein monitoring program for pharmaceutical development. Next, we will hear from Dr. Raul Rahman, research scientist at Massachusetts Institute of Technology, MIT, on integrated analytical characterization of highly heterogeneous drugs. For a complete biography on Dr. Mimi Roy and Dr. Raul Rahman, please visit the Biography tab at the top of your screen. Dr. Mimi Roy, you may now begin your presentation.
Hi, everyone. Thank you for your time and for listening in today. I'd like to speak to you about advances in the analysis of host cell proteins in drug development of biologics as well as gene therapy. I'd like to focus a little bit on how new tools in mass spectrometry have been so advancing so quickly, and how these advances can be utilized to come up with better tools for analyzing manufacturing processes. So the influence of mass spectrometry on host cell monitoring has been tremendous. The advances in mass spectrometry can be compared to advances in how our cell phones have evolved over the last decade, and there's so many new things we can do with these technology advancements. So how can we use this technology for the analysis of impurities which are coming from the host cell that is used to manufacture a biologic? So in today's talk, I'll be outlining some of the technology for doing identification of host cell proteins and also doing relative quantification using mass spectrometry. I'll be focusing first on applications that are directly utilized to enumerate host cell proteins through identity or quantity. And after that, I'll speak a little bit about a very interesting topic in the field, which is determining the antibody coverage of host cell proteins. So assessing the quality of the antibody reagents that are used in commercial release, for example. And then I'll be moving on to doing some absolute quantification of host cell proteins. So once you know which proteins you want to monitor, you can actually quantify them. And that can also be done using mass spectrometry. And finally, I'd like to end with an example, maybe a couple of examples of how one can also use mass spectrometry for root cause analysis in case there are hitches in the process development before we have a final manufacturing process. So as you all know, in drug substance manufacturing, the identification of host cell proteins is a critical piece. And mass spectrometry not only allows us to identify and give names to these proteins, but also quantify the amounts of these proteins relative to the desirable drug substance. And going on to the next slide, the typical approaches for host cell protein analysis so far have been focused on an ELISA approach, which is where you use antibodies and you take the drug substance in some kind of a form before it's completely purified, so the harvest or one of the purification steps, and you inject that into animals and you elicit an antibody response. Then you collect the antibodies, and using those host cell protein antibodies, you make an ELISA. And then that ELISA can be used in a commercial process rather inexpensively and reliably. So there are some issues with the ELISA. One of them is that there is no assessment of what the identities of these host cell proteins are. And the identities can give us some idea of safety, and they can also give us a good idea of which manufacturing process step might be an issue if things are not going well. So there are advantages of knowing what are the identities of these host cell proteins that are impurities. And since the ELISA cannot do that, what we've been doing is using mass spectrometry to assess the identity of these. The advantage of mass spectrometry is that not only can it identify host cell proteins, but it can also quantify them with respect to a relative quantitation compared to the drug substance, and also an absolute quantitation, so that you can have an absolute number and a limit for what's acceptable and not. So the other advantage is that host cell proteins can be monitored by mass spectrometry, just as in ELISA, by monitoring many proteins all at the same time. So there's a multiplexing capability that has become very popular recently. The typical approach in terms of mass spec technology is that we still rely on accurate and sensitive detection of peptides by doing a triptych digestion. So we take the therapeutic protein, and usually any protein, even if it's purified very well, has some residual host cell proteins. And these might be very small quantities, but they might still be there because they either have affinity to the protein, the drug substance itself, or they may be sharing qualities of the drug substance so that they collude in a purification step. So what we can do is take these in-process or drug substance samples and digest them and subject them to LC-MS and LC-MS. There are a multitude of methods within mass spectrometry that can be used. One can use proteomic style methods where you can do a label-free quantitation. You could do labeling and do quantitation. Regardless, you end up with a whole bunch of mass spectra of these proteins that are separated by chromatography and then measured using accurate mass and MSMS, which is your fragmentation pattern that allows you to figure out what the peptide sequence is. Once the sequence is known, we can match that with the database and figure out what protein that peptide belongs to. So in terms of chromatography, there are many options these days. There's the traditional one-dimensional chromatography. Popular methods are just doing a reverse phase on the peptides. That's something an analytical chemist is usually quite comfortable doing. So that's an option. If one goes into wanting a deeper look at what's present in the sample, then 2D chromatography becomes a really good option. These days, it's quite nice to use high pH reverse phase, referred to here as HPRP. And this kind of method allows you to do online reverse phase of two kinds that are slightly different, so you get a little more coverage through the 2D chromatography. You could also do something like strong cation exchange, or SCX. One of the disadvantages of this is that you almost always need salt. And then after the 1D separation, you have to remove the salt. And often this leads to removal of some of the analytes themselves as you go through desalting procedures. Whether you use 1D or 2D LC-MS, it should rely on, the experiment itself. If you want to find the really, really small amounts of host cell proteins, then 2D would be recommended. But if you're just trying to assess something without a very deep look, then 1D should be sufficient. One of the choices that I've found in my experience makes quite a bit of difference is choosing the database that you want to search your mass spec MS-MS data against. The MS-MS data refers to fragmentation data. This is the data that allows you to actually get an identity. And the identity, it relies on previous knowledge, on knowledge as to what proteins are present in a given organism. So if you're using CHO cells, for example, then a CHO database is needed. If you're using an insect cell line, then an insect cell line database is needed. If you're using E. coli, then an E. coli database is needed. Regardless of the database, it's advisable to add the protein sequences that you know are, in addition to the database, going to be present in your sample. This could be the sequence of the drug substance. It could be mutations of the drug substance if you know those are present. It could include possibility of searching post-translational modifications as well. While we're handling samples in the lab, we know that some of the keratin from the garments, silk garments that one may be wearing, or by touching samples without gloves, one can get keratin as a hit. So it's good to actually include it in the database so that you can identify these, and then these peaks don't get searched against other proteins, for example, and give you some kind of a score that's not reliable. There's also the addition of proteins that might be specific to a given cell line that you're using, that are specific to that manufacturing or process development exercise. And if it's CHO cells, for example, you might also want to add a rat and mouse database, a rodent database. And I would always do searches with multiple search engines. Some of the products that are out there right now are agnostic of the mass spec vendor. You can go in and do searches with the same software using multiple kinds of mass specs, and you can also do multiple kinds of search engines. And there's software available for merging all of that data. Once you have all of that data, what it looks like is that you have all of the IDs, but if you're interested in relative quantitation, then it's quite important to do some of the things really carefully. One of them is the chromatography. To have reproducible chromatography is quite important when you're comparing samples, because as we all know, as mass spectrometrists, we have suppression of signal when you have a change in gradient and different things are coming off at the same time from the column and going into the mass spec. Then the way the mass spec records the signal and the intensity of that signal is affected by coeluents. So it is really important to get reproducibility and prove to yourself as a scientist that the setup you're using is well under control. This is just an example of 3 replicates, and it's a UV profile because it's just the first step of a 2D method. And you can see here that all of the peaks are lining up quite well. And so if you are comparing 2 different samples, for example, then you would assume that the suppression from a given coeluting peak is similar in all of your samples. And so the response factor won't be inadvertently suppressed in one sample versus another due to irreproducible gradients. As we go into the second dimension, what you can see here is, even though you've done reverse phase in the first dimension, as I showed in the previous slide, using a high pH reverse phase gradient, you're seeing peaks elute in the beginning and the end of the chromatograms here. that's a bit of a trick that people use. It's using a pooling approach that has been published before. I'm happy to tell you more about that in the Q&A session if someone's interested. But basically what you can do is run your sample through the first dimension and collect, instead of collecting fractions, 8 fractions in this case, you could, for example, collect 48 fractions. And then you could pool every 4th fraction. And that way you're sampling different fractions with different chromatography profiles from the reverse phase, early eluents, eluents in the middle, and eluents at the end. And you're pooling them, and that way you're really utilizing your mass spec time. So you can see here in E and F fractions, you see quite a bit of peptides eluting in the beginning as well as at the end, and that's achieved by pooling. So these are pooled fractions, and pooling can be done in many different ways. It's a topic of its own, very interesting. But going on to how we can use these technologies, the comparison of SCX to HPRP is shown here. And even if you combine Mascot and Sequest or Bionic, your favorite search engines, and put together a Venn diagram of all the proteins that you've identified that are not the drug that's being manufactured, so they're all the host cell proteins, then you can see that depending on the first dimensional method, you get different amounts of proteins. In this case, I'm showing what I've typically seen in the past, which is that high pH reverse phase does give us a little more coverage. However, SCX gives you proteins that are not covered by HPRP. And so if you really want to do a detailed characterization of the material, it's best to use both methods. It's not a whole lot of more effort because you're just running that 1D chromatography separately and doing desalting. After that, you can combine all of the material and then run it, or you can run it separately, but you do get more IDs. So let's focus a little bit on what kinds of applications this technology can help. So host cell proteins are obviously monitored, as I explained, in drug substance, but also in in-process samples. And that's the beauty of mass spec that is hard to do with ELISA. With mass spec, you can go all the way up to the harvest and understand how the sample and the content of the sample is changing through the purification process. One of the common examples shown here is when you do affinity purification and then you do LC-MS to look at what proteins in your drug substance are being pulled down by the affinity purification. In this case, if you've taken some antibodies, let's say that you outsource to a company to make, and now you just want to enumerate what are the proteins that this antibody binds to that are present in your process. You could apply these antibodies to an in-process sample, intermediate sample, or to the drug substance. And once you have these anti-host cell protein antibodies, you can use these to bind the host cell proteins and the drug substance. You can do a pull down and immunity affinity purification. And then you can subject this to LC-MS and elucidate the identity and amounts of these proteins. You can also compare 1D and 2D to decide which approach to take with your actual samples. And you can see what kind of coverage you're getting, and that can help you decide how deep you want to go in your analysis, because deeper analysis is more effort and more resources. So if you're resource strapped, you can do 1D, but you can do a quick evaluation and compare this. So in this case, we see that there are 7 proteins common to both, but there are actually 8 proteins that you see in the 1D that you don't see in the 2D, and vice versa. So one of the issues is that when you process samples more, you do lose a little bit in the process. And so this is just an example where if you did 2D, you would get a lot more coverage. As I mentioned before, you can use this technology to follow a process. You can try to understand how to improve the purification process. You can try to understand which host cell proteins are removed at which step of a purification process, for example. And so this is showing in process steps, let's call them 1, 2, 3, and 4. And what we've done is measured the number of HCPs that we see by mass spectrometry. And you can easily see from the data that before purification starts, you see a whole bunch of other proteins. And this is 1D data. If you do 2D, you probably see, hundreds of proteins. But as you go through the purification step, you can see how these are removed. And you can, in fact, note which ones are removed when, so that if in commercial, if you inadvertently see an impurity that's unexpected, you can actually look up that impurity by doing an MSMS on that impurity peak and go back and see which process step is failing. And then you can troubleshoot these kinds of process issues. So you can see here, we're going from many, many HCPs through the process and showing very nice process control and purification efficiency. This is another step to show similar data, and you can see that as we go through purification, the number of hits and the number of spectral count numbers, or PSMs, that you get increases as you go from step 1 to step 4, and you're enriching further and further as you go down the purification process. And this is completely expected, but you could use this data to compare 2 different purification strategies and see which column removes more host cell proteins, or which column removes host cell proteins that you know are deleterious to have, for example. This is a comparison of how a drug substance, which is purified by 2 different purification processes, looks when subjected to HCP analysis by mass spec. You can see that process A has 26 host cell proteins and process B has only 1. And so if one is, as one should be, caring about patient safety in drug development, then obviously the choice should be process B, which ensures that there are very, very few, just 1 host cell protein remaining. And then one has to make sure that one can measure the levels of these and make sure they're extremely low and acceptable. So this kind of data has been obtained by a 2D LC-MS process. And what this shows is when you see only 1 residual HCP by 2D LC-MS, that the process engineers have done a terrific job with developing process B. And this process could be preferred in their decision-making in collaboration with analytics. These are just the numbers comparing HCPs to ELISAs in ppm. So as I mentioned before, ELISAs don't tell you what is there, but they're pretty good at assessing the total quantity of all HCPs that the ELISA is able to measure. And one can compare these and get an idea of, how the 2 different orthogonal methods of measuring HCPs track, and they track very nicely together, so it's nice to see that. We can also compare manufacturing of the same process done at two different sites. Process engineers and process validation engineers and people who work with transfer of processes are well aware that making a biologic at a new site is not a joke. It's quite difficult to get exactly the same process. And good analytics, high resolution analytics using mass spectrometry can be so informative to understand the difference in manufacturing of the same drug substance using the same process at two different sites, where there might be some differences in the equipment or in the operation details and so on. So this is an example of comparing two sites, and what you can see is the two sites are really quite comparable because they both can give you similar HCP HCP content in the drug substance. And you can see that site A has 13 HCPs, site B has 14 HCPs, but 12 of them are common. So we would call this quite comparable. And you would also have data on the identity of these proteins. And as I'd mentioned before, if you map out which protein is removed at which step, you can actually troubleshoot this and come up with an absolutely identical process if you had to. One of the examples of using mass spectrometry that is my favorite in this field is actually not just using host cell proteins directly as the analyte and understanding what they are and how much of them are present, but to really have that conversation with the commercial strategy and the quality groups in the departments where these processes and testing are developed to see if the ELISA is the preferred method for commercial release because it's commercially a very well-established technology, and it's inexpensive, it's reproducible, it's highly sensitive. Then the most important application of mass spectrometry actually falls in understanding the ELISA and understanding what are the host cell proteins that are recognized by the antibody, the anti-HCP antibody that is being utilized in the given assay. So for this, the regulatory authorities expect commercial development and commercial release to assess the antibody coverage of the host cell protein antibodies being used, and to really make sure that the coverage is not low. Typically, I've seen 30 or 50% coverage to be considered quite low by the regulators, because basically that's saying that only half of the host cell proteins that are actually present are recognized by the antibody. So we're blinded to the other half, and that's not a very good analytical method. It's nice to have something above, I would say about 70%, and the higher the better, because this is ensuring that the antibodies that we use are highly sensitive and selective for the impurities that we're trying to make sure are controlled and monitored. This is a traditional methodology, and for those of you who are mass spectrometrists, I'll go through it a little bit in detail so that if it's of interest, then you can think a little bit more about it. So on the left panel is shown a 2D gel. So this is no different than doing a 2D LC-MS, where you have an enumeration of all the proteins that are present in a sample. And so with antibodies, what you could do is compare the proteins that you see in a 2D gel in your material. So let's say you're using a harvest material to inject into animals and elicit an antibody response. And then you're going to use those antibodies as your reagent for the ELISA. Then to measure the coverage of these antibodies, you need to compare the total amount of protein in the harvest to the proteins in the harvest that are recognized by the anti-HCP antibodies. So essentially, the former is assessed through the 2D gel, which is on the left, and the latter is assessed through the 2D Western, which is in the middle. And the way the assessment is done is by overlaying these two images and seeing which of the proteins that are seen by the 2D gel that are indeed present in the harvest are recognized by the antibody and showing their presence in the 2D Western. And by taking a fraction of the numerator being those peaks that are seen and recognized by the antibody, and the denominator being the total number of host cell proteins or spots that are seen on the gel, you can get a percentage, which essentially is your coverage. Now, there are some issues with the traditional approach. Running a 2D gel is actually technically more difficult than running a 2D LC-MS once you have the mass spec technology in place. Matching spots between a 2D gel and a 2D Western is actually quite difficult, as you could see in the image in the previous slide. Reproducibility is one of the biggest issues with 2D gels, and you always have to go to your favorite RA to do this, because not everybody can do it so well. So it's quite subjective, and as scientists we want to have objective methods. We want to have reproducibility in our measurements regardless of who did the work. So these are some of the limitations. The other limitations are that you just lose the peptides, so if you have something that's clipped and there's a little peptide that might elicit an antibody response, that may not be very safe for a patient. Well, you'll miss it in a 2D gel. You're also going to lose coverage of the proteins that are very close to the DS because the DS is overwhelming in its signal. Now, a 2D LC-MS approach helps a lot with that. Quantitation is also difficult, and what's a little bit frustrating to the mass spectrometrist who knows all about identification of proteins using mass spectrometry is that this kind of technology gives you no identification information at all. And that's a little bit of a disconcerting fact, given that we have all of this technology to do identification. The way we use mass spectrometry to do coverage is, instead of doing a Western, we use a column and affinity purification. So we have the antibodies on the column, and we pour the harvest through the column and evaluate the harvest, which is equivalent to looking at the 2D gel. And we evaluate the eluate, which is what didn't bind the antibody column. You could also, you can do the flow through, which didn't bind the antibody column, and you could do the eluate, which is what bound the antibody column and then was eluted out. If you do just an analysis of the eluate and the load, you can get a very nice estimate of antibody coverage. And we've done these comparisons for a column followed by LC-MS to a more traditional method with 2D Westerns, and we found the numbers to be not very different, but the nice thing here is we have the identities. So one of the mysteries that we saw a while ago was that when you look at the eluate, you start to see proteins that are not in the harvest. We all know that mass cannot be created or destroyed, and so the proteins that are measured in the eluate must be going in the harvest. And what we realized is that because it's an affinity capture, there is a concentration effect. So there are proteins in the harvest which are very low concentration that may not get identified using even the 2D LC-MS method. And when you go through the affinity, they get enriched and you start to see them in the eluate, but you don't see them in the load. And what we've come up with as a strategy is to include those proteins in the load so that you're not underestimating or overestimating the antibody coverage, because it's really important to get that estimate correctly. You can use these kind of antibody assessment experiments to assess which animal, species A or species B, as shown in the left, is better to produce antibodies in. You can even compare individual animal sera using these methods. And you can also compare, let's say, a commercial antibody kit that comes with the antibody that is used in the kit. And you can make your own in-house reagents and see if the in-house reagent is really much better. There are quite a few good commercial antibody kits these days, and there's no reason to prefer an in-house antibody reagent if the commercial kit is giving you very nice coverage already. However, it's nice to have that data to back up your decisions and to make good decisions for commercial assays. So we've been using these kinds of technologies also to do assessments of in-house versus commercial antibody reagent production decisions. Going on to the second big topic for today, we also want to talk a little bit about absolute quantification. So in the previous examples, we talked about comparisons. So you say 2 proteins here and 10 there, and you can make an assessment. You don't need to know how many nanograms per mil of each protein is present, because that's a lot more work. However, if you need to, you can do that. And you can do that using multiple reaction monitoring, typically using a triple quad instrument. The way this is done is you have the same sample prep workflow, but instead of doing a discovery type or a proteomics type approach, you do a triple quad approach where you've already elucidated what proteins are in your sample, and you build an MRM assay, which is a targeted quantitative assay. Through the triple quad, what you can do is separate the target peptide, do fragmentation in Q2, and then Q3, you can choose the transitions, the fragment ions that lead you to the signals for each transition going from Q1 to Q3, and you can develop a target peptide multiple reaction monitoring assay. In our hands, we've been able to monitor 400 proteins in a complex sample at the same time. So this is very doable. What you can do also is synthesize the peptides and develop standards so that you can have these made, usually outsourced. And you can have these outsourced and made as heavy labeled entities, and you can have them at quantified amounts. So now you know when you add these and spike them into your sample, what signal in your MRM corresponds to what amount. And then you can use these to elucidate the amounts of the unknown peptides. By unknown, I mean the unknown amounts of peptides, not the unknown identities, of course, because you have a targeted assay, you know the identities. After assay optimization, the final assay will have all of these transitions, like you can see in the bottom left, where each peak actually stands for a given peptide and therefore a given protein. And you can have hundreds of these in the modern triple quads that we have. If one worries about the quality of the assay, one can qualify these assays and validate them as well. And this is just an example of how stable these triple quads are. We have internal standard peptide 1 and internal standard peptide 2 that are run along with the samples for about 170 samples run over several months. And you can see how beautifully consistent the signals are, and the CVs are well below 10% for the first peptide and below 10% also for the second. So you can get quite accurate assessments. The spiking of the heavy peptide, as I mentioned, allows you to do absolute quantitation, and you can compare the intensity of the unknown, in this case, the red peak, compared to the blue peak, which is a spiked in heavy peptide, and therefore you can measure the unknown red and come up with protein concentration in nanograms per mil. In a process development scenario, as well as in cases where there are surprising peaks in a purification process, you can figure out which step in the purification or even in the harvest is contributing to a higher level of a given impurity. That might be an investigation if you're looking at lots of DS and the impurity is changing in amounts depending on the lot. As I'm showing in the next slide, you can do the MRM or a relative quantitation and correlate the mass spec intensity signals of these peptides with all of the process parameters that go into the purification. For example, there might be a pH in a given step. There may be, a column volume, a buffer, could be many process parameters. And you can do a multivariate correlation analysis as this shown here. And you can try to find those process parameters that correlate with showing up of an impurity. As shown in the next example, we've chosen one purification step parameter on the right, and you can see the blue signal actually goes up for lots 5 and 6. So we can tell that lots 1, 2, 3, 4, 7, 8, 9, 10 are okay, but there is something going on with lot 5 and lot 6. And so if we can take all of those parameters and then do the HCP analysis using mass spec, we can actually find exactly the identity of the proteins that are showing up high due to a change in the process parameter. We can use this to correct the process. We can use this to understand exactly which process step is an issue by looking at the identity of the protein, having mapped the processes with these HCPs. So there's a lot of possibilities for developing robust processes so that one doesn't have any issues in commercial manufacturing. And also if there are site-specific issues or there are issues with, cells in harvest having growth problems or other things that people do see during process development, then one can correct them before building a final robust commercial process. So I hope I've convinced you that mass spectrometry has developed to the point that it's a fantastic tool for analyzing all the way from harvest to purification steps and to the drug substance or drug product. And these approaches are powerful in identification as well as quantification, relative or absolute, of host cell proteins, and that these are quite powerful, and they're powerful compared to antibody-based methods, but also they can be used with antibody-based methods. So it's not a this or that, but it's really a combination of power in both approaches that can yield the best of results. The 2D LC-MS approach provides much better separation because it spreads out in 2 dimensions the signal that one is going to see by mass spec, resulting in a deeper look at the impurities if this is desired. And the affinity purification mass spec provides a very nice assessment, a reliable assessment, along with the assessment of quantities as well as identities of HCPs. And this kind of assessment of antibody coverage is very, very important for regulators to see, to know that the commercial assay has been developed and is able to have good enough coverage to have confidence in commercial release. Proteins with poor antibody coverage can also be identified using these methods, and the commercial assays can be supplemented accordingly if needed. The MRM assay is a wonderful tool for doing very quantitative assessments of host cell proteins. They can be used to specifically monitor given impurity, to also support root cause analysis during process development. Thank you very much. And if you have any questions, I hope I can answer them to your satisfaction in the Q&A session. Thank you for listening.
Well, good morning, everyone. Thanks for the introduction and this this great virtual meeting. I'm glad to be able to present our work. So I'm Rahul Raman. I'm from the Sasi Sekran lab, and I'll be talking about the integrated characterization of biopharmaceuticals. And this work has been largely in part from a thought leader award that Ram Sasi Sekran, who's a professor at MIT, got from Agilent in really trying to move the field forward and move the needle in terms of using the Agilent equipments for to come up with some very important workflows for characterization of biopharmaceuticals. So when you look at biopharmaceuticals in terms of the biologics, so clearly, they are obviously complex molecules, and the way we think about it in terms of the complexity is coming along these two axes. So the first axis is really looking at you're going from your starting material, which is the cell line plus vector, through the upstream to the downstream to get your drug substance, drug product. So clearly, a lot of parameters going from the starting material heterogeneity in terms of the cell state, the genetic stability, culture conditions, the upstream, downstream process steps influence some of the key product attributes, and definitely establishing these relationships between the starting material process to product is a very important aspect of the biopharmaceutical development. And the other axis that we focus on is going from these product attributes to quality in terms of both from an efficacy standpoint with respect to the antigen binding, the effector functions, and specific aspects of safety in the standpoint of immunogenicity, aggregation, and any other issues with the product that could potentially lead to adverse events. So clearly looking at this in the context of process to product and product to quality is the way we want to address understanding, deriving these relationships to address the complexity in biologics. And a key part of doing that is through developing integrated analytical workflows. So when we look at integrated analytics, so clearly there are obviously many ways of defining it, but from our perspective, we kind of look at it again at two levels. So as you know, from many regulatory considerations, there is always a push to develop orthogonal methods and integrated methods. So our perspective is when you take any attributes in terms of a specific structural attribute, the way we would like to think about orthogonality or integration is to have multiple measurements that are pertaining to the same attribute at a given heterogeneity layer of the mixture. So one example is if you're taking at N-terminal modification like pyroglutamate, so you can use the peptide mapping as well as an orthogonal chemical approach like NMR to have correlative readouts to pyroglutamate content. And in another example, when you look at the complex glycosylation modifications and you release your glycans and you want to characterize them, they're again having both like a mass signature as well as a chemical signature from linkage and linkage abundance from NMR can provide very important orthogonal correlative measurements for a given attribute. So another way of looking at integrated analytics is to obviously look at the same attribute across different layers of heterogeneity of the molecule. So in this context, clearly, if you take glycans as an example, and I'll walk through this a little bit more in detail in the later part of the talk, is you can obviously define glycan structure and glycan modification at the level of the intact molecule, like if you take an antibody at the level of the intact antibody glycoforms. and you can define it at the next layer, which is sort of the glycopeptides. That is, once you fragment the antibodies into peptides and glycopeptides, and then at a third layer, you can look at it as if you release all the glycans from the antibody and characterize them. So depending on however you define these layers as top down and bottom up, the ability to correlate very specific aspects of the glycan modifications across these different layers become another important aspect of this integrated analytics. So some of the advantages of looking at analytics and these data cross correlations from orthogonal methods is it really overcomes the limitation of any single method in terms of sensitivity and resolution to sort of accurately measure an attribute and how to define the sort of the process to product and product to quality relationships by just relying on sort of just a single measurement. So it really sort of removes that bias and gives a more robust way of defining these attributes in order to sort of measure these key relationships. So given that we had these sort of perspective and with all the successes that our lab has had in characterization of complex molecules such as polysaccharides and also looking at development strategies in taking the molecules to sort of rapid development path. So Agilent actually recognized Professor Ram Sasisekharan and the Sasisekharan lab to provide this Thought Leader Award. And an important part of the Thought Leader Award was obviously to sort of come up with important workflows using Agilent's state-of-the-art instrument to sort of characterize monoclonal antibodies. So as many of you know, monoclonal antibodies have heavy chain, light chain, and their source of heterogeneity from a product standpoint can come from a whole host of modifications, including glycosylation, deamidation, backbone modifications, disulfide shuffling, and a whole host. And each of these modifications provide unique signatures when we look at both process to product relationships as well as product to quality relationships. So when we set out to sort of implement these objectives in the Thought Leader Award, so the first thing we wanted to do is obviously lay this out into some sort of a map where we look at the attributes at various levels, from biophysical, biochemical to the protein sequence structure and backbone modification, and actually have what are the kind of heterogeneity that we are expecting to see, and what is the impact in terms of at what level from the process to product or the product to quality that these attributes would impact, and what are some of the analytical methods, both from the standpoint of orthogonality and measuring across the different layers of heterogeneity that we need to have in place to sort of measure these attributes. So one example, again, is if you take glycation or deamidation, they're very important signatures of cell culture. Well, like glycation is a very important signature of cell culture medium. And it's a pretty good signature for process fingerprints. And it can also look at it from the standpoint of stability and downstream and the overall product quality. In a similar manner, if you take deamidation of asparaginins, again, they play a very important role in structure function depending on the location of some of these residues at the key interface of epitope, paratope interfaces. And again, they provide important signatures of both downstream process and formulation as well as product quality. So when you go to more complex modifications of these antibodies, so clearly glycosylation is a pretty complex modification because the glycans are very heterogeneous, both from a chemical as well as from a topological standpoint, and they play a very important role. So given the nature of these glycan modifications like terminal sialylation or core fucosylation, play a key role in sort of regulating the half-life of the antibodies, regulating effector functions such as antibody-dependent cellular cytotoxicity. So sort of monitoring these glycosylation as a function of both the process product and product quality axes become very important. So having the right set of tools to sort of develop these correlations become very important from the standpoint of the integrated characterization of these biopharmaceutical products. So with this sort of a map of what we need to measure and what are the kind of capabilities that we need. So we worked with Agilent, and they sort of gave us this really cool set of instruments, which at that time were sort of the state of the art, which includes the 1260 bioinert HPLC and another HPLC with a very specific functionality to do SEC with a multi-detector array, including light scattering, to look at the aggregation properties as well as high molecular weight, low molecular weight species. And of course, the coolest toy, which is our favorite, is the 1290 Infinity 2 UPLC linked to, I think what was at that time really the state-of-the-art acute of MS, because I think at that time when we got this instrument, only 2 other labs, including a test lab, had it. So, and we are pretty pleased with this instrument in terms of the capability in analyzing the biopharmaceuticals as it will become evident in the later part of the talk. And the other instrument which we've used more from a discovery standpoint is the whole assay map Bravo, which is an automated liquid sample handler, which enabled us to do a lot of microtransfection purification, to do a rapid screening at the level of intact antibodies, to sort of simultaneously derive some critical attributes across hundreds of designed antibodies that would really help in accelerating and supporting our antibody discovery efforts. And of course, along with all that, the key supplies from the columns in terms of all the various columns required to do the analytics. Again, Agilent was generous to offer. And the other important relationship in terms of Agilent from this thought leader is, it's not that it was just, you have these tools, you have these columns, and so you sort of go do your stuff. But they were actually working with us to see, what are the shortcomings in these tools? How can they improve the software? How can they improve the column chemistry? And so there was a sort of a pretty good sort of relationship in the context of having a collaboration with them to really sort of make sure that these solutions are robust for industry standard biopharmaceutical characterization. So the specific example we chose in terms of looking at the characterization using these tools is to look at the NIST antibody. So the NIST antibody is actually supplied by the National Institute of Standards and Technology. It's actually a humanized monoclonal IgG1 kappa antibody expressed in a murine culture. And this has actually undergone industry standard upstream, downstream purification and formulation. And it has also been extensively characterized using other analytical instruments, including thermo, waters, and so there's an extensive amount of reference data available in several publications for this antibody. So it provides us with an sort of an example to both validate our workflows as well as sort of compare what we've gotten with what some of the other instruments have offered, and also provide this feedback to Agilent to just make sure that whatever technology that we are developing is sort of on par with whatever has been sort of published already about this antibody. So in terms of the antibody characteristics itself, again, it's been pretty well documented in terms of where you have these modifications, both in terms of glycation, deamidation, oxidation, which are the specific residues that have all these properties. So again, with this wealth of information, you have a pretty good set of reference data for us to sort of compare and validate our analytical workflows. And as I said, there are multiple labs with multiple instruments that have analyzed this, and all these results have been published actually. So what I wanted to do is obviously summarize all the things we have done, but focus very specifically on a particular aspect of the integrated analytics with respect to the glycosylation modifications that we've done with the NIST antibody. So we have obviously developed workflows and validated them against the reference data for the intact antibody analysis, the heavy chain, light chain subunit analysis, and then the full peptide mapping and backbone modification analysis, size variants, charge variants, and the glycosylation analysis. And so this is sort of extensive data that we are actually putting together with the help of Agilent into a compendium to provide sort of an additional reference for this data using all the Agilent instruments, so which will hopefully be published fairly soon. But one of the areas that I wanted to focus on in today's talk is to give you an example of how we look at the integrated analytics for characterizing the glycosylation. So as I mentioned earlier, the example from our perspective that we wanted to showcase here is how do you look at glycosylation at the different layers of heterogeneity, which is at the level of the glycoforms at the intact antibody level, to sort of the post-translational modifications at the glycopeptide level in terms of the specific glycosylation site that gets modified. And the third level is once you release these glycans, sort of how do you characterize and quantify them, and how do you develop some of these correlative relationships between the glycans characterized at these various levels actually. So with the NIST map, there is only one N-linked glycosylation site, which is the most common glycosylation site across most of the IgG1 antibodies, which is at the 300 asparagine position. So this slide talks about how you actually do the IDES digestion to sort of separate the Fab from the FC. And given that the FC is the one that has the single N-linked glycosylation site, you kind of get the glycosylation at the intact domain level in terms of looking at it in the context of the LCMS to look at the various glycoforms and sort of rank order what are the top glycoforms as a part of this. And this is again consistent with what has been reported with some of the other mass spec instruments in the literature. So the next level is looking at the actual glycopeptide that has the asparagine 300. So here, instead of just cleaving the antibody, breaking just the antibody into the Fab and FC level, you do your typical peptide map like trypsin and these other sort of proteases to sort of break them down into individual peptides and glycopeptides, and actually do the LCMS analysis of these peptides, glycopeptides in a way you would do the peptide mapping. And the Mass Hunter software of Agilent is again pretty good in sort of picking up these glycosylated forms of the glycopeptides and actually coming up with pretty accurate annotations of the glycan topologies based on the mass signatures of these glycopeptides. So this represents the layer of at the glycopeptide level for the analysis of the glycan signature. And then the next level is, of course, looking at the released glycans. And so we release the glycans using PNGSF, given this N-linked glycosylation site. And we sort of label the glycans. We've tried different label forms, including 2AB and some of the other labels. And we, again, use a Hylic LC column to do the LC and followed by LCMS to get a detailed read on the types of glycans that have been released from the antibody. So as you can see, these sort of represent different types of samples that have been analyzed, but the information contained in this is the same, which is what are the distinct glycoforms or the glycan structures that are present as a part of the post-translational modification of the antibody. So what we are sort of in the process of doing is to sort of develop these correlative measurements where we kind of rank order these glycoforms and actually see in terms of their relative rank ordering at the level of the intact structure, at the level of the glycopeptide, and at the level of the released glycans, is sort of the overall ranking maintained, which kind of gives the confidence of the most abundant species to the lowest abundant species and the distribution of these glycoforms at one level. And it also, we're also trying to calibrate it broadly in terms of what is the speed of the analytics required to sort of get this ranking. Like obviously the IDES followed by the intact glycoform analysis is the fastest method. And clearly the glycopeptide analysis from the data processing is sort of a relatively slower method where the released glycan is somewhere in between. So the reason for also doing this is if we want to build these type of correlations for different biopharmaceutical products, so that if you want to develop in-process control, we can still rely on the fast method because we now have enough of a robust understanding that the rank ordering of these glycans would be preserved, looking at it across the various layers of analysis, and whatever changes or differences that we can pick with either a process change or something that is happening as an in-process control, we can try to use some of these fast methods to actually detect those and develop those as sort of important methods for the process development and the process product correlation. So these type of correlative analysis is what we want to sort of build in addition to developing the workflows as a part of the integrated analytics platform. And the other types of correlations which we can also build is, even looking at these backbone modification variants, where when you look at charge variants specifically with respect to the acidic, main, peak, and basic variants in terms of just looking at them through the column analysis, and you look at it more distinctly in terms of backbone modification with respect to the cleavage of the C-terminal lysine or deamidation, you can see a pretty good correlation between looking at the charge variants to the backbone modification, again, where the information contained in the backbone modification is richer because you have more specificity, but the speed at which you can sort of do some of these things at the higher levels with respect to looking at the acidic and basic variants, you can look at it from the sort of the LC analysis and sort of make up these correlations. So the main point is we are obviously doing with the Agilent instrument what is sort of known in the industry in terms of looking at all of these modifications as critical quality attributes. But in addition, we are also trying to develop these important correlations, which will give a good indicator of the product drifts and other things that happen during process development or during stability analysis, to give this additional sort of information for sort of improving the product during the product development. So the last few slides, I just wanted to focus on the other aspect that I was saying, which is the assay map Bravo, which we have obviously used for the sort of a lot of our antibody discovery efforts. So the assay map Bravo is actually an automated liquid sample handler that I think Agilent had sort of acquired and developed it quite well. So it has multiplexing capabilities in terms of looking at key quality attributes such as yield, aggregation, and this it works off of a 96 well format that's compatible with the sample handler to the LC system to the LCMS system. So that way you have cross-platform integration going from the sample handler all the way to the analytical instruments for these multiplex readouts. And it also helps us permit sampling a large grid space of our designed antibodies. And rather than just looking at them as displayed SCFVs, we can actually make them in sort of these micro transfections into full antibody structures and look at the critical quality attributes. And it is also quite modular, and it has, we've used it for discovery, but it can definitely be, the scope of the application can go towards looking at process development, looking at building these relationships between process to product and the product to quality. So the typical workflow that we've developed for looking at our antibody design and discovery is you have these design constructs, you make them, and the bottleneck right now for us is to actually do the 96 well transfections, which is still manual, but that can very easily be automated through a lot of these robotic transfection processes. And then the assay map Bravo actually has a protein A micropurification. And once you do the micropurification, you actually have a sort of a very small amounts of these purified antibodies, which we can rapidly analyze for multiple attributes, including yield and expression, looking at the SEC for like any aggregation, and doing all the quality checks with the at least getting the intact mass and mass analysis with the LCMS to do sequence verification and things like that. So in that context, this assay map Bravo has really been a very important tool as we look at not just biopharmaceutical characterization, but also discovering and developing new antibodies. So I think this is sort of where we are with all the Agilent instruments that we've gotten. And as I said, we're in the process of writing some of these things up into technical app notes and compendiums. So we'll plan to publish all of this pretty soon. But I think this has been a very good journey so far in terms of working with Agilent, and we sort of look forward to continuing this collaboration as it represents a key step towards developing robust workflows for the characterization of biopharmaceuticals. So with that, I'd like to acknowledge, of course, Agilent for the Thought Leader Award, and Ram Sasisekharan, who is the recipient of this award, and the members of the Sasisekharan lab who are involved in this work, Chengu, Vidya Subramanian, and Andrew Hagas. So I thank you for your time and attention, and happy to take any questions. Thank you.
Roy and Dr. Raman for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located at the far left of your screen. Now let's get started. Our first question is going to be for Mimi, actually. How can we increase throughput of sample analysis as well as data analysis?
Oh, that's a wonderful question. I guess we've talked about it a little bit, not in my talk, but in the recent one, that, you know, the assay map Bravo is one of those examples of a 96-well platform where you can do sample prep, you could do 1D cleanup, you could also do 2D cleanup, you know, do the first dimension of chromatography on the assay Bravo instead of using a column, and use different elution buffers, for example. And so that's an example of just increasing the throughput by sheer automation. One can use different mass spec technologies as well, like the multiplexing of assays using MRM. That's a nice way to increase throughput instead of following one or two proteins, you know, following many of them at a time. For data analysis, we really like the mass center quant option where you can do automated data analysis, and you can you know, really increase the throughput of your data output.
Thank you. And another question for you. Are these synergies in detecting HCPs and product-related attributes?
Oh, yeah, definitely. I think someone's got their mute button undone. I hope you can hear me. So you can look at, sorry, can you repeat the question one more time?
Oh, yeah, absolutely.
Distracting.
Yeah, it's okay. Are these synergies in detecting HCPs and product-related attributes?
Yeah, absolutely. So the same mass spec methods that may be used to look at post-cell proteins, for example, identification of new proteins, it's all based on fragmentation or based on some knowledge of PTM and so on. And the same kinds of technologies can be used to look at the product itself and a lot of the modifications that a product might have, some of the vulnerabilities of the product, which is something we really want to get a good handle on, and, you know, really assess critical quality attributes early on. All of those can be done with very similar technologies to what we're talking about in the post-cell protein detection piece.
Thank you. And one more question for you, Mimi. What are the aspects of these technology that are most exciting for the future?
The most exciting thing for me is really combining all of these technologies and taking them to that next level of process analytical technologies through in real time, if possible. So imagine a world where you would do, lot release with, a really powerful peptide map that could follow product quality, as well as host cell proteins, as well as, some residuals all in one assay. So mass spec has the capacity to do that.
Cool. Well, thank you so much. Have a couple questions for Raul. What are the potential areas for improvement of Agilent analytical tools?
Yeah, hi. So, yeah, I think the Agilent tools, it's been sort of developed very nicely for the integration aspects, like I said, from the sample handler to the analytics. So one area which I know we worked with them is clearly sort of how to look at the data analysis software to make them sort of more compliant for quality control, specifically, looking at monoisotopic masses, looking at other areas of improved annotation for glycosylation when you compare with some of the other mass spec instruments that are there. So it is more to sort of work with them on some of these software improvements. And the other areas have also been with the column chemistry, where I know they're again constantly trying to improve some of the chemistries for like better resolution of charge variants and some of the other things based on how that performs compared to some of the other columns for which this NIST data has been published. So the good thing is we've been actively, as a part of this award, we've also been actively working to give some of this feedback so that they can improve this to the next generation in terms of improving both from a quality compliance as well as a resolution, all the things that can be done.
Thank you. Another question for you. What advantage does Assay Map Bravo offer for screening for antibody quality?
Yeah, so the good thing about the Assay Map Bravo, in a way it's set up for the protein A, is again, rather than looking at just fab fragments or SEFVs, in a way you would look at the libraries. One rapid read you get with these sort of microtransfections, and I think they can definitely be scaled with robotics going from 96 well plates to sort of multiplexing formats. That you now have the entire antibody with its properties in the system. So when you're looking at yields, when you're looking at sort of this aggregation, you kind of get an early readout on some of the candidates that may sort of pass in your antigen binding affinity binning buckets, but they may sort of have a very diverse profile in terms of expression or some of these other aggregation properties. And we've even tested that when you go from the microtransfection to large scale, that some of these properties do sort of get amplified. So you sort of get early readouts at the entire intact antibody level when you do the screening to sort of help pick good candidates with a better development path towards a therapeutic.
And last question for you, Raul. For complex modifications like glycosylation, what is the extent of characterization that is needed, and how does one look at orthogonal tools?
Yeah, so that's like obviously a big question in the industry too. So it depends on, again, so there's a trade-off between resolution and speed when it comes to glycosylation. And from a regulatory perspective, what becomes important in terms of looking at the quality and the safety efficacy becomes an important point. Like if you take monoclonal antibodies in these typical manufacturing CHO cell lines, they have pretty standard glyco profiles. So you don't need to go into the in-depth in terms of the chemical linkage and some of these other properties unless you're engineering the glycoforms for a very specific effector function, like you're sort of removing the core fucosylation for improved ADCC. So depending on the kind of information that you're looking for, you have to go to that extent of analysis. Of course, the more analysis you have, the more confident you are of the product, given that this is a pretty complex modification. But where it becomes more apparent is if you're changing the expression cell line for some reason where you get a better yield, or you're going to say like other antibody formats like bispecific antibodies or FC fusion proteins. So there you could see a lot more variability in the glycosylation compared to what is typically known for monoclonal antibodies in CHO expression systems. So that's where you would like to go more detailed into sort of orthogonal tools like having enzymatic degradation profile to get at specific linkages for salic acid, or even combine them with some analyses like NMR, which I don't think has been done routinely, but it offers a lot of opportunity because NMR gives a pretty good read on the chemical linkage profile of glycans, which you can also sort of add as an orthogonal tool to some of these LC and MS systems, depending on, that if you're really going to a new platform, new molecule, that you need to characterize them more extensively to sort of mitigate risk from a safety to the actual quality of the molecule.
Thank you both for your answers. Do you have any final comments for our audience by chance?
No, I mean, thanks for the opportunity. I think this is sort of the first time I'm doing this this webinar. I think it's worked out very well with the pre-recordings, and thanks Mimi for your wonderful talk. This is a talk is just listening to in this session, I hope this was a very useful session for everyone who was signed on to it.
Well, it was a pleasure to have both of you, and thank you again so much for your time today and your important research. We really appreciate you. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand through June of 2021. Until next time, goodbye.
Thank you so much.
Thank you.